be brave if you're brave. I'll be brave, but only if you're brave. And it could be just you and me. We'll be family. Just wait and see. So I will fight if you'll fight. Yeah, I will fight, but only if you'll fight. Oh, we can make it through this like sailors in a tempest. Like sailors in a tempest together. And it could be just you and me. We'll be family. Just wait and see.
Good evening, everyone. Welcome to tonight's Lung Cancer Awareness Month version of the Lung Cancer Living Room. Thank you all for being here, everyone in the room, those of you joining us online. I hope you're sitting down somewhere cozy in your homes, ready for a great evening tonight. For those of you who don't know me, I'm Danielle Hicks, and my role here at the Foundation is to oversee patient services, patient support programs, anything that has to do with touching the patient, from education programs like this all the way down to one-on-one navigation. So welcome, welcome, welcome. We are so excited to have you all here tonight. We're very excited to have Dr. Amy Moore, who most of you in the room know, some of you online may not know. Amy is our Director of Science and Research for GO2, and she'll tell you a little bit about who she is and why lung cancer matters to her. Dr. Christl is also here to talk to us tonight about our exciting topic. I think I didn't mention it, but it's talking about how drugs are developed and how they ultimately get to the patient, which is a little bit different than what we normally talk about. But I'm kind of excited to be able to let you guys understand what that process looks like, why it's important, and what role you can ultimately play in helping to get these drugs to what we call the bedside or to the patient. So with that, we're going to go into the program. I do want to give you guys a little bit of background on Dr. Christl, who's here tonight. She's the Executive Director of Global and Regulatory and R&D Policy, JAPAC Lead and Global, formerly with the FDA, and an expert on drug development. So really, we couldn't have anyone better here coming to talk to us about what this drug development looks like and how we ultimately get some of these amazing drugs to you guys, the patients. But before we get into the sort of topic of tonight, I'm going to ask you guys both to give us a little bit more background on who you are just as people and why lung cancer is important to you. So I don't know, Amy, if you want to start, and then we'll come back towards me.
Sure. So I am trained as a basic researcher. My PhD is actually in virology, so I worked on viruses before moving into cancer research as a postdoc. I actually worked on leukemia, but then when I left the research bench, I became part of some statewide cancer initiatives in Georgia. So a lot of what I did there was focused on building infrastructure. So we helped recruit top cancer researchers to academic institutions throughout Georgia. We outfitted their laboratories, we funded them, and it was all with the goal of moving the needle and trying to improve patient outcomes, but it was still very abstract. And so life brought me to California, and when I started looking for job opportunities, happened to see the posting for the Dario Lung Cancer Foundation, and it was very similar to what I'd been doing in Georgia, just laser focused on lung cancer. And for me, the attraction was, everything I did in Georgia was abstract. I never touched the patient directly. And this has been the most fulfilling job I've had because I get to work with all of you. And it reinforces the urgency of what we do daily. And all of our lives have been touched by cancer. My life has been touched by lung cancer. My uncle by marriage just passed away earlier this year. He was diagnosed as ALK positive and had a very short run, unfortunately. One of my good friends from high school, her sister fought ALK-positive lung cancer for 10 years, young, prime of her life. And so we all know the stories, we all know the people. And for me, this is, something that's bigger than myself. And so I try to every day come in and do what I do to make a difference. And I'm motivated by trying to equip and empower researchers with the tools they need to change lives. Ultimately, it came down to the decision for me, kind of doing that internal assessment, what did I want to do with my life? I said, I'm not going to be the one who cures cancer, but I want to help those who will. And this is how I came to do that work. And so that's what I do every day.
Thanks, Amy.
Okay. My name is Leah Christl. I am also a scientist by training. I'm a molecular microbiologist by education. My mother tells stories. I was always that scientific person. My favorite word was why. I did science experiments in my house in the bathroom cabinet and frightened my sister on multiple occasions. And I was just always one of those people who always wanted to know why, who wanted to look into how things worked, why they happened, what were the answers around things. So that's really been a guiding principle in my life. So I went into science. I originally did environmental research, working on hazardous waste degradation, trying to remove environmental contaminants that then can cause disease in humans and animals from the environment, and approached public health that way. After I graduated from grad school, I worked at a university and taught classes and had my own lab. And then I had the opportunity come to me to go work for the Food and Drug Administration. So I said, why not? Something I didn't know how to do. And again, the other question of why and how can I help? So I was at the Food and Drug Administration for a little over 16 years, working in various places within the FDA on drug development in different areas, beginning in over-the-counter drugs, and then ultimately moving into biological therapies, and then specializing in biosimilars, which I think we'll talk a little bit about as well. After 16 and a half years at FDA, I wanted to take that next step. And then looked at working for a pharmaceutical company. So I went from being in an environment of regulating drugs, reviewing them, helping to bring them to market, to then moving to a company that was developing those drugs. But my job in my company is focused on what's called shaping the regulatory policy environment. So as a former regulator, I know how these organizations work. And so within my company, I am then advocating to help shape that regulatory environment, to make it more favorable for drugs to come to market, to have better understandings, to open up the pathways for things like innovative clinical trial designs and targeted therapies, and be able to move that regulatory process with the science so that we can bring more drugs to patients.
And so with that, I'm going to jump into and you two as well, like if you want to kind of go back and forth with one another, I'd appreciate it too. But walk us through just like a basic process for getting drugs from the bench to the bedside or from the lab to the patient. What does that process look like from a timeline perspective, from a cost perspective?
Okay, I could start. Feel free to jump in. So there's really not a set timeline to bring drugs to the market. It really depends on the drug, the disease, what we know about it, if there's more fundamental research that needs to occur before candidates can be identified to then move forward. But a lot of studies show it's anywhere from 6 to 12 years from that bench to the bedside to be able to bring drugs to patients. But one of the important statistics to understand is that 12 to 14% of drugs in clinical trials make it to patients. They're the ones that get approved. So there's many, many candidates that don't make it through this whole process that we'll talk about. So really, it's only, again, that 12 to 14%. It's an expensive proposition. There's estimates that range from $1.5 billion to $2.6 billion to develop a new drug and bring it to patients. So there's different ranges for different types of drugs, but for an innovative therapy, it can be up on the high end range of that $2.6 billion to be looking at innovative therapy. So there's a lot that goes into it with that. So in terms of that development process of bringing a drug to patients from that bench to bedside, it does truly start at the bench. And again, it's what we know about the disease that's being targeted, what we know about the science, what we know about what's referred to as the mechanism of action. So how does that disease work? What's happening in the body? And from there, scientists can be in the lab and try to look for places where they can affect that process and identify candidates that can disrupt what's happening in the body in terms of that disease progression. So for oncology therapies, we're looking for disrupting, growth of tumors. Is there a way that we can cut off blood supply? Can we, decrease replication? Can we, what can we do to make sure that they're not hiding from the body's own immune system? There's general therapies, as folks know, from chemotherapy that's systemic. We're now seeing a lot of advances into what are referred to as targeted therapies. And again, a lot of that comes from what we know about the disease. The more we know about it, the more we can look at those targeted therapies. And so where you have in lung cancer, the EGFR, the ALK mutations, the KRAS mutations, to be able to look at that and say, well, what do we know about how that's different from some other mutation or where there isn't the mutation, and to be able to then look for things that we can do in the laboratory to disrupt that process for those types of things. So there's a lot that's looked at in the lab. There's what we refer to as screening of candidates. They do that in the lab in a non-clinical type of setting of looking at different cell lines that are grown, where you can have cell lines that are taken from patients, where tumors are removed and then those are grown up and tested in the laboratory. You can have cell lines that are made in the laboratory as well, of trying to create what's referred to as transgenic models. You start with a transgenic cell line, and then you can look at, testing and are there responses from those cells.
So when you say cell line, can you explain to people what you mean by a cell line? Like what is that?
Right. So a cell line. So again, your body is made up of cells. So for oncology, for cancer, your tumors are also made up of cells. And so within the laboratory, what they try to do is have those cells, those individual cells, and then be able to grow them so that they have material to work with in order to do that. And so they have to find the right conditions, and once they do that, to be able to grow them, they can keep these, essentially cell lines growing to continue the testing in the lab and to continue their work that they're doing.
Yeah. Some of you might be familiar with the book called The Immortal Life of Henrietta Lacks, who was a patient with cancer, and they harvested cells from her and been able to propagate them kind of indefinitely in the research lab. So that's one of the things, some cells are able to do that, others are, more primary. They aren't able to be maintained in culture as long-term, but we can do, still some studies with them. So there are different types. We typically grow them on dishes. Some are what we call adherent, so they stick to the dish. Others grow in suspension, so we have to know kind of the right formula, the right culture media to grow them in. So they require different nutrients and things just like we do. So each cell line might be unique and specific and have very
Okay, Good segue. So to continue with drug development, so moving from the bench, the next stage is typically going to be animal studies. So drugs are then that are viable candidates to be able to move forward into further testing will typically be tested on animals first before moving into any sort of human trials. So looking for mostly for safety issues there from a short-term type of standpoint. There's also long-term animal studies that go on, usually strangely enough, carcinogenicity studies, other types of studies that would be from long-term exposure to understand that those continue through that, the life cycle of developing a drug, because those are more longer-term studies based on exposure. But there are short-term, more acute types of animal studies of looking for immediate safety issues in terms of toxicity from exposure to the drugs before they're ever moved into humans. So once they have that data, again, if they think that it's viable to be moving forward into human trials, it then does move into clinical trials.
And so I'll just interject at that point, again, with alchemy, we have a couple of animal projects going on to create both PDX, patient-derived xenograft models for EGFR, as well as ROS1 mutated lung cancer. So that's where patients can donate some of their leftover tissue or pleural effusion to create these models. And it fills an important need, as you heard. You've got to kind of go through these steps to get the drug from, the cells to the animals to then testing in humans. So, that's part of the pipeline and part of the need that we try to fulfill.
Right, and with those animal studies as well, before starting clinical studies, you can start to get an idea of the dose that you would want to use, the amount of drug that you would want to use. So there's safety and tolerability studies, essentially, that they'll use higher doses, and they see where they may get a response, or maybe when you start to see safety issues, and you could choose a few doses then, based on what you're seeing in those studies, to then move into clinical trials, that you have some of that information. As science is progressing, you can actually get some early information about potential efficacy of the drug through these types of transgenic types of animal models that you would have, where you're taking cells from that lab and injecting them into animals, and they develop disease, and then you give them the drug and see do they respond or not respond. So it's not always predictive of the response in humans, but it can start to give you some information about the viability of that candidate moving forward, or is there something highly concerning there that you need to do more testing on before you ever look at human exposure. So once you pass those stages and you have something that you want to move forward, and again, things don't work, things happen. So this is where you start to see, candidates dropping off that lead you to that lower percentage, making it through that whole process. You move into the clinical testing phase. And so traditionally it's referred to as phase 1, phase 2, and phase 3 testing for clinical trials. And so phase 1 is obviously the beginning, and those are going to be fairly small studies in humans. Typically, they are just with drug. It's not a placebo. It's not an active comparator. It's not standard of care comparison. What you're doing is the first time that you're putting that drug in humans, and so you're looking at how humans respond to that. Sometimes phase 1 studies, depending on the drug, can actually be done in healthy subjects, because you're looking at safety response in a lot of cases, not necessarily efficacy. So for some of the drugs you can do that, for some drugs you can't do that. But again, you're looking for those types of responses. Typically in those phase 1 studies, you're looking not just for safety, but you're starting to look at exposure in the body, how that response is going. If it's in patients, you might get a little early information about how they're responding to it from an efficacy standpoint, and then they choose the doses that then they want to move forward into further clinical testing. In the phase 2 studies, that's where we really start looking at the drug's efficacy as well as the side effects. These tend to be larger studies that are there. You're looking for circulation in the body, how that's working, are there any safety issues. There you may or may not have a placebo control, an active comparator, or comparison to standard of care, because you are looking at early efficacy information. So you'll have some more data that's coming out of it. If things look favorable there, you then move into what's called phase 3 clinical testing. And these are what are referred to from a regulatory standpoint as well and adequate controlled clinical trials. And these are the trials that are the basis for approval. These are the pivotal safety and efficacy trials on which a regulator will make a decision about approving a drug. So this is really, where the rubber hits the road. So these are going to be large trials. These are always going to have some sort of a comparator with them in terms of either placebo-controlled, active comparator, or standard of care of comparing the efficacy. So this is where you have what we talked about before, with looking very carefully at inclusion-exclusion criteria. There's rules around those clinical trials about how they're run, what data is collected. There's a lot of interface with the regulatory authorities globally as to what the endpoints are. What is going to be your evaluation for efficacy? And that's referred to as the primary endpoint. And that's going to have to meet a certain standard. So to say that you saw within a certain percentage some sort of an improvement, either in comparison to placebo, standard of care, or some other drug. You can look at the safety profile. You can also have what are referred to as secondary endpoints, and this is where some of the information might come out about those subpopulations, folks that might respond in a different way, or if you're looking for folks that respond differently if they have a certain mutation. So those are those secondary endpoints. They won't meet that same type of statistical test all the time, but you want to get data from those subpopulations as well, and that's why they're secondary endpoints. It's not a pass-fail for that trial the way that primary endpoint is, but it's very important data. And sometimes if you fail on the primary endpoint, the information you get from those secondary endpoints shift your focus in terms of drug development, and then you develop new trials to be able to look at, well, who responded, and can we do a clinical trial for those responders then to then continue that drug development that's there. So I don't know if you have any other.
You're doing great.
So I have a question. Well, two questions actually. So one, what are usually the timelines for each of these phases? How long typically does a phase 1, phase 2, and phase 3 trial take?
There's not necessarily perfect set timelines for them, because a lot of it is you're moving into phase 2 and phase 3. It depends on the disease that you're studying and how long it might take disease to progress. If you're looking for overall response, if you're looking for progression-free survival, if you're looking for overall survival. So overall survival is certainly the longest endpoint, and so that trial, if that's the endpoint, and that's typically what it is that you're looking at ultimately for oncology therapies, that can go on for a long time. In the oncology space, however, looking at overall response and progression-free survival and other types of endpoints can sometimes allow what's referred to as accelerated approval in the US. And that's based on an endpoint that wouldn't get you full approval, but they're seeing that there's response that's there, and so they want that drug to come to market to be able to be available to patients while they're collecting that other data about does it work or does it not work. And so a lot of the oncology therapies go through that process. So you're able then in a real world setting to treat more folks. You still have your clinical trial ongoing. And if you have accelerated approval based on what's referred to as a surrogate endpoint, so it's not for full approval, there's a requirement that they conduct confirmatory clinical studies to do that. And so there's a certain time frame that they're supposed to do it in there.
We've seen that a decent amount in lung in the last few years. Go ahead. Go ahead, Kat.
I just have a quick question. Do either of you have a wish list of what you would like to change in this space, in the clinical trial space? Like you just wish that there was a different...
There's a wish list. There's a lot. No, I mean, I think, we've touched on a lot of the issues of cost, access, innovative trial design. There's lots of still room for improvement, even though progress is being made. I think we all wish we could get, better drugs developed faster and get them to patients. So, but how do we go about that? I mean, I don't pretend to be the expert in that arena, but yeah.
Yeah, I mean, I think from my standpoint, I'm a big communication person, and when I was at FDA and working on biosimilars, we had an internal facing component but an external facing component, and being able to figure out how people needed to get information, and at FDA, how we interfaced with patient advocacy organizations and what we could do in terms of communication and helping to empower patients to be the best healthcare advocates for themselves that they could. And having a family member or someone else, it's a complicated space. So, my wish list always is to be able to develop those tools. I do that at my company now as well to help support that, to give people the information that they need to really navigate this space. And so that's really my wish list, is to have more transparency.
So this has been great. We thank you all so much for all your questions and keeping this really interactive. And of course, a huge thank you to Leah and Amy for coming out tonight and chatting with us about this very important topic.
Oh, I'll be brave if you're brave. I'll be brave, but only if you're brave. And it could be just you and me. we'll be family. Just wait and see. So I will fight if you'll fight. Yeah, I will fight, but only if you'll fight. Oh, we can make it through this like sailors in a tempest. Like sailors in a tempest together. And it could be just you and me. We'll be family. Just wait and see.